technique. The Nobel was awarded recently, and single particle reconstruction is going to be the structural biology technique of choice, as everyone can see from the increasing share of structures deposited. When we do single particle reconstruction, what we do is we take EM images of a field of our particles, and we obtain something close to a 2D projection of the molecule's density, as you can see here on the right. Obviously, we need the 3D structure, not just these 2D projections. So to get back to the original, we have to reproject lots of these particle images back from different angles to obtain a 3D volume. So the problem here is that we don't know what angle they were projected from originally. So what we do is to make a best initial guess, to align our particle images to projections of that guess, and then we iterate back and forth, hopefully improving the structure each time, although that's not necessarily the case. So unfortunately, as this image shows here from Bob Glaser, cryo-EM images are super noisy. This comes from a variety of noises. Shot noise is one of the principal contributors, but heterogeneity, including radiation damage, is also a significant contributor. And what this means is that you end up incorporating noise, a lot of noise from each of the images every time you reproject. And as you can see from this example from Shaw Sherris lab, if we leave the noise in the structure, we just get the wrong result. So here is an alpha helix being shown in its best fit into data which has been reconstructed containing lots of noise. And the true position of the projected structure is shown on the right. And you can see that the best fit will be completely wrong if this noise is left in. So how do we distinguish our noise from our signal when they're convoluted together in this way? It turns out that the only way to do this reliably, that anyone has come across so far at least, is to do two entirely independent reconstructions from half data sets, and then to compare them together and filter maps accordingly. Currently, people typically compare them with the Fourier shell correlation, the equation for which is shown up top. This is developed by Herritz and Van Heel. And then they weight them according to CREF, which is effectively the FSC that would be generated if it was against a perfect noiseless volume. This was work from Rosenthal and Henderson, and that's derived from the FSC. So what we do is we do split half sets, make a series of reconstructions, iteratively calculate the FSC, and then eventually we combine these and weight and low pass filter for interpretation, because the noise in the images increases with increasing resolution. So the highest resolution that we are able to work with will be the point at which the noise overcomes the signal. So this all works very well for some very well behaved samples, but unfortunately this isn't crystallography where everything's sitting in a nice array and our particles are free in the ice, unfortunately, and not held in place in the crystal. We're not in crystals anymore. We get flexibility, and what this does is that it moves, say, for instance, the heads of these ducks here. When we average these together, it gives us the hideous many-eyed Thulu duck instead. We also have problems with variable occupancy, as you can see here. Some of these have heads, some of them don't. And this can sometimes be sorted away, but sometimes it can't, and this is dependent on the sample. The end result of all of this is that we end up stuck with noise that we're trying to exclude from our reconstruction. It's coming in from these particles, noisy background. And once again, the results can be effectively impossible to interpret. You can see that this is all popcorn up here, and this is a factor that's bound to a ribosome. So people have historically tried to handle this by calculating FSCs over small windows within the total reconstruction, and then calculating local resolution rather than a global resolution in exactly the same way that they would if they were working with the entire map. And then to remove this noise and allow them to interpret and work with their data, they simply low pass filter within these small windows in exactly the same way that someone might filter globally according to the FSC. There are problems with this approach that the window choice in terms of size affects the outcome. You can see that varying the box size here from say 10 to 25 significantly changes both the predicted resolution and also that at the peripheries where we have relatively sharp edges, we start getting quite significant differences in the calculated local resolution just based on where things are in the reconstruction rather than the actual signal to noise ratio. And most significantly, this approach won't weight regions to retain as much signal and to remove as much noise as possible. Instead of weighting something nicely, we simply truncate it entirely at some point. So what we did was we set out to make a filter, which we called LAFTER, that handled this better by looking at this as a signal to noise ratio in real space as opposed to resolution in Fourier space. So we take an input voxel and we weight all of the input voxels at each resolution shell according to the probability that they are still part of this noise distribution. And then we truncate each voxel in real space at the highest resolution that its signal is still greater than the maximum of the noise distribution. So if you think about when you open a noisy map in Chimera, you play around with the slider in order to remove the dust. And the point at which you set it is usually just where you can't see the dust anymore. And this is a similar kind of thing that we're doing here, except on a massively increased scale. So when we look at this with synthetic data, which we've generated, which is deliberately noisy, here's a true signal, and here are noisy maps at different resolutions. We can generate reconstructions, which exclude features that are effectively beyond the resolution at which we know that there is signal. And this also works extremely nicely when the signal to noise ratio is deliberately kept excessively low in synthetic data. So here we're able to recover clearly recognizable signal in the LAFTER maps from these noisy maps, from synthetic data when the FSC is at two commonly used global cutoffs. Obviously, this is something that you'll never see in experimental data, but it's worth noting that other local resolution filters cannot do this as they simply truncate and don't weight. So Reliant and Blockfield, for example, wouldn't be able to handle such weak data. When we actually look at experimental data, that's all that we really care about, obviously. LAFTER performs extremely well as a local resolution filter. Because it's working on SNR, we get the lower SNR signal showing up at lower contours, as you can see here in comparison to the experimental map, and all of the popcorn in the experimental maps is gone, and we still retain the high resolution features at relatively higher contours. And you can also see here that LAFTER retains the same high resolution signal that matches the models without noticeable degradation between the two maps, the LAFTER map and the experimental map. They're almost exchangeable. So we know that if we don't account for local resolution when we're working with our final combined output maps, we get lots of uninterpretable noise in our final reconstruction. But what about during the refinement process? I said that we were weighting and filtering here as well. Isn't it bad to include the noise here too? Well, yes, of course it is, actually. If we generate synthetic data with known local resolution, as you can see here at the top, and then look at the FSC within masks, you can see from the SFC curves within these masks that when we do the standard FSC weighted reconstruction process, we're getting overfitting. These curves are drifting to the right in regions that are kept at very low local resolution in comparison to the true situation where we just do an idealized reconstruction on the left. So the sensible solution would seem to be to do exactly what we did for the final reconstructions, for the intermediate reconstructions. We locally filter during refinement instead of globally filtering at the end. We weren't able to just use LAFTER for this because we need to maintain the grayscale for the refinement program, and importantly to maintain the independence between the half sets, because otherwise we can't calculate our FSCs, and we also don't know what's noise and what's signal. So here I can show you that if we did a standard windowed FSC filter, like block filter, which is the one I showed you earlier, between two white noise half maps, we actually get a significant FSC correlation right the way out to Nyquist. This is the situation without any kind of filtering. But we were able to generate an SNR filtering approach, working forward from the same kind of SNR approach that we were looking at with LAFTER, that would maintain independence between these two sides. And we've called it side splitter because it works for the split sides. So when we apply side splitter during the refinement, in this case a rely-on refinement, but it should in principle be compatible with any refinement program, to this synthetic data set, you can see that we reduce this overfitting problem in the FSCs. We push these curves back to the left in the comparison to here, although we can't get rid of it entirely. So is this actually any use experimentally? Well, yes. When we apply side splitter to experimental data that we know has a very severe propensity for overfitting, we're able to suppress that overfitting. So here you can see in a mice cell that's from a membrane protein, we get lots of spurious radial features due to misalignment in an FSC weighted refinement, whereas in a side splitter refinement, we're able to suppress these almost completely. This also works quite nicely when we have a series of mixed conformations. So here we've deliberately left a series of mixed conformations in an experimental data set. There's less overfitting in the mixed region, which I've circled here. You can see in the side split, this is significantly weaker. And the actual output map that we get at the end of this is better with side splitter. So you can see here, for instance, we have a complete section of main chain, whereas here there is a breakup. And the same goes for mice cells. Here, this is a membrane protein, and we have all of these spurious features within the mice cell, relatively high local resolution, which disappear completely in the side splitter refinement. And there's also an improvement in the map. Here you can see that there's a relatively continuous chain in the side splitter refinement, whereas we get broken up data again in the high resolution regions of the standard refinement. So the take home message from this is that global anything doesn't really work for any averaging approach in cryo-EM because the samples vary in real space, and therefore you need local approaches to handle them. Local filtering and subtraction are going to be incredibly important in future. High quality real space filtering will allow us to move to iterative subtraction of density to handle multiple conformations and occupancies, because we'll just be able to work from convergence at each time. LAFTER and SIDESPLITTER are GPL. The source code is available on GitHub, and binaries that have been pre-compiled are available through CCP-EM, thanks to Colin. SIDESPLITTER is run quite happily from a wrapper as a hook from Rely 3.1, and that's work thanks to Takanori. Thank you very much to my funders, to anyone who has helped, to the organizers for asking me to talk, and to anybody who has bothered to listen to me prattling on for the last 15 minutes. Thank you.
Thanks very much, Chris. So there are some questions coming in. Chris, can you turn your video on?
Did that work?
It probably will in a moment. So first of all, well, this is not quite working.
There we go.
There's a question from Uma Mathur-Jan. Which version of Rely were LAFTER and SIDESPLITTER integrated into?
That's Rely 3.1.
And how much does the resolution improve after running LAFTER and SIDESPLITTER in a typical scenario?
So it will depend entirely on how much how large the regions of low local resolution are. So if you have extremely large regions of low local resolution, you should expect to see a significant improvement. The PFCRT dataset is the prototyped for this kind of thing, I think, and we saw that go down to 3.4 angstroms, which is similar to the published data, which had subtraction of the micelle. I think, generally speaking, the approach is worthwhile whenever you would see significant differences in the local resolution. If you're working with a very rigid particle, then it's probably not worth bothering with. But if you're seeing any reasonably large regions of local resolution, then it's worth trying.
It's an interesting question here. Will using SIDESPLITTER run the risk of removing information you might want, such as the level of the membrane in reconstructions involving nanodiscs?
So it depends. This is sort of philosophical to a certain extent. It depends whether or not you want the best alignment on the parts of your reconstruction that agree or not. I would argue that you're better off getting the best possible alignment on the parts that agree, and then looking at other parts either by sorting or by subtraction. But it is certainly a philosophical difference of approach. If you are quite happy working with relatively poor angles where you see things a little bit better at, say, 7 to 10 angstroms resolution, then that's up to you. Yeah.
And then the last question I think we've got time for is a controversial one, difficult one to answer, so answer carefully. What's the SIDESPLITTER advantage compared to non-uniform refinement in cryo-Spark?
So my understanding is that non-uniform refinement requires 4 reconstructions to be carried out, and that therefore the actual signal to noise of the maps, if you see what I'm getting at, is going to be half. I haven't done a direct comparison, and to be honest, I think that the approaches are both converging towards looking at things in a similar way, but time will tell.
Okay. Thanks very much, Chris. I think we'll leave it there and move on.
Thank you.
It's a great talk.